Alright, so tonight, by midnight, you need to turn in your narrative of neural signaling. And essentially, I just want you to describe to me what happens from one neuron releasing neurotransmitter and being received at the dendrites. So start there, right? Neurotransmitter activating neurotransmitter-gated channels. Tell me about the different types of channels, the synaptic potentials, EPSPs, IPSPs, processing in soma, decision point, how does the signal get transmitted down voltage-gated channels down the axon, right? What happens at the axon terminal in terms of releasing the neurotransmitter, and then how does the neurotransmitter get taken out of the synapse, which I'll talk about today. Sam.
So the neurotransmitter and the neurotransmitter opens the neurotransmitter.
It does, for sure. All right, so this is that whole process right here, right? I want you to describe all of the stuff that happens, right, from here all the way through to release and getting it out of the synapse.
Are there any link requirements, like how long to make that mine's trying to be great?
No, it doesn't matter. I mean, you can be concise if you want to be verbose. I don't really care. The point is for you to internalize this and be able to sort of tell it as a story. I think if you try and memorize all these bits and pieces, I think it's complicated and abstract, but if you can tell it like it's a story, this is what happens when a neuron gets activated, right? That's fine. You can hear Caroline and then Anna.
Do you want the violin stuff?
Yeah, you can include the fact that there's saltatory conduction and that it's only degenerating with the notes. I think that's probably important.
How is this graded? How is this graded?
How is this graded? I'm going to give you feedback on it. So if you're missing parts, you're going to get it back with feedback. Which is why I want to get it tonight. I'm trying to turn it on tonight. Okay. All right, before lunch, that's my point. Just to get it back to you. Okay? All right. All right, so okay. So I'm going to come over here and I'm going to draw a diagram. So there are four processes in neural signal, right? Four processes in neural signaling. Input happens at the dendrites, right? And there's a part of the neuron associated with each of those four. Input happens at the dendrites. Processing happens in the soma. Transmission, the action potential, happens in the axon. Output happens at the axon terminal, right? And along with that, there are important channels, right, associated with each of those. So there are neurotransmitter-gated channels at the level of dendrites and soma, right? There are voltage-gated channels starting at the axon hillock and along the axon and at the axon terminal. 3 neurotransmitter-gated channels, 1 for EPSPs, sodium comes in, 2 for IPSPs, potassium goes out. This is positive, if it leaves, it gets more negative inside. And chloride is negative and comes in. Right? We have 3 important voltage-gated channels, 2 of which are used for the action potential. Sodium comes in for the rising phase up to positive 30. At negative 40, potassium opens. When does sodium open? Sorry. That's important. Negative 50, which is threshold, which is the decision point. We get to negative 50, boom, we're opening the sodium and action potential takes off. Rising phase up to positive 30. Here, this is what's sorry. Forgot I was going to draw this. Nice. What are important voltages that we need to know.
The threshold and
Okay, threshold of -50. What else? -70 is the resting potential. +30 is the peak. -40 is where potassium opens. -75 is the refractory period, right? So, bring the right? So we have inputs, neurotransmitter gated. Sodium comes in, creates an EPSP. Sodium only comes in for a short period of time, right? And then then why does it go back down? Right, it closes. It closes in the pump. The sodium-potassium pump is constantly working, right? It's pushing 3 sodium out, pulling 2 potassium in. 3 sodium out, 2 potassium in for every ATP molecule, okay? We have an IPSP where we let potassium go out. We can have another IPSP, right, where we let chloride in. All right. So how do we get to -50? One EPSP is not going to do it, right? Can't.
Is it spatial? Do they build on top of each other?
Yes. Summation, temporal and spatial summation. They happen close in time before the pump can pump out the sodium. They add up, right? So if we have one, and then before the pump can pump it out, we get another one. And before the pump can pump that out, we get another one. And if we hit -50, all of a sudden, the voltage-gated channel is going to open, and we're going to let so much sodium in that we're going to go all the way up to +30 before that sodium channel closes, right? And that's the rising phase of the action potential. And that sodium that comes in spreads out along the axon down to the next node and activates the next voltage-gated channel to keep the signal going. Behind it, what happens at -40? Voltage-gated potassium channels triggered, short delay from -50 to -40. It stays open longer. So now that the sodium is closed, potassium is leaving, right? And that's our falling phase all the way to -75.
So after you hit +30, then you're going back down? Is that
Yes.
okay?
Because we're not letting any more sodium in, and the only thing that's happening is positive potassium ions are leaving. So that's going to bring the charge back down because all that potassium is going out. So much potassium goes out, it comes all the way back down to -75. Anna Clark.
So at the beginning, when you have the first sodium in, that small red one right there, that's demonstrating that it wasn't enough, like it wasn't enough.
Didn't reach threshold.
Okay.
Right? So action potentials, the decision is yes or no. That's a no. That's a yes. The decision point is, did we add up to -50 or not? If we get to -51, which is right here, nothing happens. right? If we shoot past and go up to -45 with our inputs, what's going to happen? Does it matter that we went past? No, because when it hits -50, it triggers it, it opens, and then we just go. Just got to get to that threshold, which is what it's called now, right? Hannah.
So I think I understand sodium and potassium's relationship, but I'm not sure how chloride works into the equation.
Yeah, so we'll talk about chloride more today because we're going to talk about some neurotransmitters, and GABA is a major inhibitory neurotransmitter, and GABA primarily works to allowing chloride to come in. Okay? It's just another, there's two, you got two inhibitory signals, but only one excitatory, essentially. Okay?
And it has its own channel? Does it have its own channel?
Yes, it's its own separate channel.
So because sodium opens earlier than potassium, that's why you have like the rising and falling.
That's why rising happens before falling, right? And remember, as the sodium is coming in, it's making it more positive inside, and that's causing potassium to want to leave more. Because when it's still negative, as long as it's below 0, right, potassium is kind of getting held in because it's attracted to the negative charge. But as soon as it goes past 0, it just wants to exit very quickly, right? And that's what causes the falling phase. So then we have all the sodium inside and all the potassium outside. We got to reset all those forces. So we have the refractory period, where the sodium-potassium pump is resetting, is restoring, right, those concentrations. And until they're built back up, we can't send another signal. Because if we haven't built them back up and we open we get to -50 and open the sodium channel, sodium's already inside. It's not built back up. It's not going to do anything. So this is no action potentials here until we get back to resting potential grace.
So I read about the absolute and the refractory period. Is that something you want us to include?
Yeah, that's a little bit more detail than I'm probably looking for. So the thing is, it's like there's a period right here where there's not any, the forces aren't there, right? And as the forces start to be built up, that's called absolute. Cannot be a
And that's because
action potential.
the sodium channels are already open, is that correct?
If a signal comes in and opens them, in the absolute, nothing can happen because there's not enough sodium outside to make anything happen. As we build up the forces, you enter into this phase called the relative refractory period, which there's sort of enough ions back and forth that you might be able to send a signal, right? But it's harder. We're not really going to get into that. That's beyond the scope of this course. Just treat those two together as refractory period. We're not going to see anything until we restore the forces. Okay?
Thanks.
Yes. All right. So over here now, right, we have, this is what I've drawn up here, right? And I'll take a picture of this and post it on the other side. So now we have all of our different numbers that are important, right? So everything on this slide, everything here, that pretty much summarizes synaptic and action potentials. The only thing that's not up there, is the exocytosis process and recapturing the neurotransmitter. So exocytosis, you have the action potential coming down the axon, going in one direction, right? And it gets here to the axon terminal, and there's a new voltage-gated channel. A new voltage-gated channel that's a calcium channel. So the voltage of the action potential opens the calcium. Calcium comes in, right? Calcium comes in, and calcium is going to cause that vesicle to fuse, right? And we talked about that specific mechanism, that the vesicle is snare complex holding the vesicle, waiting for calcium. Synaptotagmin is just hanging out, waiting for calcium to activate. Calcium comes in, activates synaptotagmin, which is going to fuse those plasma membranes and allow the neurotransmitter to dump out of the vesicle into the synapse. So the important things about exocytosis: voltage-gated calcium comes in. Okay? Snare complex is holding the vesicle docked and ready. Calcium activates synaptotagmin to open the vesicle, release the neurotransmitter.
And this is the end of the cycle?
Yeah, this is the axon terminal, the output, the fourth process, output.
And it starts over again?
Yeah. So that neurotransmitter is going to go over to the next neuron and bind with the receptor, and we can have another EPS beat if it's glutamate in the cycle. Okay? All right. Neurotransmitter. So how do we get neurotransmitter out? Remember, we want like discrete signals, on-off signals, right? We don't want the neurotransmitter to stick around a long time in the synapse. We have three different types of mechanisms. The first I already mentioned is reuptake. Think about reuptake as recycling. Put those mnemonic devices together. Reuptake is recycling. It's taking the neurotransmitter back up into the terminal to be reused. Neurotransmitter chemicals, we have to build those. We have to make those chemicals. And so, that takes energy and nutrients, and so if we can reuse them, that's a good thing. Glial cells. Remember we talked about a bunch of different types of glial cells? What are some different types of glial cells we talked about? Microglia. Someone said astrocytes. Yeah. Yeah. And we talked about oligodendrocytes in the central nervous system, which are myelinating, right? So they're surrounding these neurons, and particularly the oligodendrocytes, but also some of the astrocytes, will absorb the neurotransmitter, inactivate it, and dump it in the blood as waste. It's sort of a go-between. The glial cells, the support cells, sort of a go-between to sort of get rid of the neurotransmitter. They also secrete special enzymes, two of which are called COMT, that breaks down dopamine, and norepinephrine, catecholamines, and monoamine oxidases. It's going to oxidize monoamine neurotransmitters like serotonin. This is catecholamine oxidizing enzyme. And here we see acetylcholine. Acetylcholine is primarily broken down by an enzyme. It's not these two. It's actually called acetylcholine esterase. And those enzymes essentially break the neurotransmitter so that it no longer can fit and bind in the receptor, so it inactivates it. Yes?
So would you say the molecule is broken down?
Catecholamines, norepinephrine, dopamine. We'll talk about that family of neurotransmitters. All right, good? 3 methods. That goes in your narrative. How do you get the neurotransmitter out of the synapse? That's the last thing that has to happen so we can send the next signal. Great? Awesome. Okay. Again, just another summary slide. All right, got to move on. So far, the types of channels we've talked about allow ions to go through them, and those are called ionotropic channels. And ionotropic channels can be opened by different things. They can be opened by neurotransmitter for an EPSP, IPSP, or they might be opened by voltage or the action potential. They're both ionotropic because they allow ions to flow through a pore in a selective direction, selective ions in selective direction. So, these are important neurotransmitter-gated ionotropic channels that we talked about. We also have important voltage-gated ones. right? So when that neurotransmitter sits in that receptor site, it causes the pore to open and allows movement of those ions. And they move because of the forces that are built up. Concentration and electrical forces. Right? We actually, the sciencey way to say that, right? is the channel changes its conformation. When the neurotransmitter sits there, it like moves, changes its conformation, and that opens the pore. All right. When the neurotransmitter detaches, it goes back to the closed shape. So it's only open when the neurotransmitter is present for a very short period of time. All right. Ionotropic channels can also be opened by voltage. So this is an example, I think, of a potassium channel here. Yes, so when this is negative, below a negative 40, it's a negative charge and it's like holding this channel closed. There's a positive charge on the channel, negative charge inside. But when this, you think about this, I wish it didn't say positive, I wish it said negative 40. But it's really sitting at negative 40, right? So as long as it's below that, it's attracted. But as soon as it gets above that, it repels it. And when it repels it, it forces the channel open and allows that potassium to leave. Just trying to help you understand, like, it's easy to understand the neurotransmitter is like a lock and key, how it's, when it's sitting there, it opens it. This is how voltage can open those channels. All right. Now, that's one whole class of neurotransmitters, or neurotransmitter receptors. Okay? And the big thing that I'm going to talk about today is that neurotransmitters are chemicals. It's just a molecule that has a specific shape. They are not inherently excitatory or inhibitory or anything. It's just a shape, right? It's what the receptor does when that shape binds to it that creates the action, not the actual chemical. The chemical turns on the receptor. The receptor is excitatory or inhibitory. Okay? All right. So we can sort of talk about two general classes of neurotransmitter receptors. Ionotropic, that are going to generate potentials, movement of ions, and metabotropic, also called 7TM receptors, because I draw my little what am I drawing here? The lipid membrane, right? With the water-loving heads and the water-not-liking tails, right? So metabotropic receptors aren't channels. They're actually strands of protein that go in, and they start on the outside, and they go in and out of the cell 7 times. So it goes 1, 2, 3, 4, 5, 6, 7. It ends up on the inside. And a lot of times they're just shown kind of as a solid shape, right? It's fine. But they're really like form this kind of integral signal. And when neurotransmitter binds to the outside, right? So here comes my neurotransmitter. And it binds, it's going to now, on the inside, it's going to release a thing called a G protein that is what we call a second messenger. First messenger is the neurotransmitter. Now it's going to cause release of a second signal inside the cell.
How does it go in and out of
Oh no, I didn't design it. It's just the way it was meant, right? Sometimes you'll see these referred to though as, sometimes sometimes you'll see these channels referred to as 7TM instead of metabotropic, because that means 7 transmembrane. Sometimes you might read that in something, so no, that's a metabotropic receptor if I see 7TM. And it's because it goes through 7 times. Okay?
So the neurotransmitter is actually like following kind of that path.
No. It just binds here, and this changes its shape.
Okay.
But it doesn't open a channel. When it changes its shape, it pops off this G protein on the inside. Right? And that G protein can go do things. It's a powerful signaling message. Okay? All right. So, metabotropic receptor binding to that causes the G protein, and there are many different types of G proteins. You're not going to be memorizing different G proteins, but so you just need to know G protein is a second messenger signal. It's released, and then it goes and does things inside the cell, right? So what can it do? Well, it can pop off and go open an ionotropic channel from the inside. So it could trigger a potential from the inside of the cell. In general, think about ionotropic channels are discrete signals, right? They're potentials. They're either excitatory or inhibitory, or they're action potentials, right? Metabotropic effects change the excitability of the neuron. They modulate the activity of the neuron. They're regulating if that neuron is highly excitable or not excitable. So for instance, if we wanted to make it easier for this system to work, right? What if we had a metabotropic signal that's going to open a channel and allow some sodium to leak in, and we change the resting potential from -70 up to -60? Now it's going to be easier for EPSGs. They got a 10 millivolt head start. It'll be easier to excite that cell. You understand how that works? How that can modulate? If we open an inhibitory channel, if we allow potassium to leave or chlorine to come in, we can make it harder for that neuron to get activated. Now we might lower the resting potential. So through these actions, we can actually change the resting potential inside the neuron. And usually, this sort of internal binding with the G proteins lasts longer. It's not like the neurotransmitter. Neurotransmitters kind of like kiss the receptor and release. They just like in and out. It's this very short signal. These are sort of longer lasting sort of binding. It can also come over here and say, hey, I'm going to shut you down. Even if neurotransmitter binds on the outside, I'm not going to let you open. Right? So it can prevent signals. So metabotropic channels receptors are very powerful because they change the ability of neurons to fire. Right? They modulate our activity. They not only can they act on ionotropic channels, they can also send signals to the nucleus to cause the neuron to make more stuff. Make more neurotransmitter, or make more ionotropic receptors and stick them in the cell membrane. Right? So second messengers can act through a series of cascades, if you will, to cause changes in gene expression and make things. And all of that, the things they're making, again, changes the excitability of that cell. If I now put more sodium neurotransmitter gated channels, I say, go make 10 more of those and stick them in the membrane. That's what the second messenger said to do. Right? That's going to make it that much easier for neurotransmitter to activate it because we've added 10 more things that they can activate. Good? All right. All of this is leading to these things, modulatory things. All right. So I mentioned there's this thing, a kinase, there's a cascade here, a pathway. And that happens because we get one receptor, when it's activated, it actually can release more than one G protein. It might release 10 G proteins. Okay? Those 10 G proteins, in this case, adenylate cyclase, are then going to activate, each one of those 10 might activate 10 kinases. cyclic AMP. So now we have how many molecules activated? 100, right? 10 more times 10, 100, right? And then those 100 might then, each one of those 100 might go phosphorylate or change 10 proteins. So now we've affected 1,000 proteins in the cell. Right? So this cascade, this series of chemical activations allows for an amplification of the signal so that one neurotransmitter can produce a big effect inside another. Yes?
What we're talking about now is what starts that process, right?
What we're talking about now in metabotropic channels affects that process. It modulates that process.
Okay.
Ionotropic channels do that. Right? Metabotropic channels affect it.
So do we need to include the metabotropic in our...
No, this is beyond the narrative, right? The end of the narrative was where I stopped with neurotransmitter. Okay? This is new stuff about neurotransmitters and changes and receptors. All right. There are some special connections in the body that happen infrequently. And so you can actually have receptors that are connected to each other where there actually isn't a synapse between the cells. And these are called gap junctions. And these are ionotropic channels that when the inotropic channels open on one side, it opens on the other. So essentially, this is like a wire. There is no chemical selectivity. There's no neurotransmitter in a gap junction. And so if this cell gets activated, then this cell automatically gets activated and handed off. Because remember, electricity is not selective. It's on. So when this is on, this comes on. Gap junctions are important for things that we want to all happen together. So we find gap junctions, for instance, in the cardiac cells of your heart, because we want the four components of your heart to all contract at the same time, right? We need them all to contract at once. So if we use gap junctions, when we stimulate them, the electricity goes everywhere, and the whole ventricle or atrium of that contracts at once. So they're pretty rare. They're found in rare situations. It's not a normal signaling mechanism, but they do exist. Yeah.
So most of the time it would just be like empty space between neurons?
Yes, synapses. Yes. All right.
So it's called gap junctions, but it's when there is no gap.
Yeah, that's kind of fun, right? It is. So remember, we talk about in signaling, right? When we have a synapse, we talk about the neurotransmitter side that's releasing as the presynaptic, and this is the postsynaptic. And then this one would have a presynaptic and postsynaptic, post at every synapse, right? So everything we've talked about so far has been postsynaptic receptors, right? Receiving neurotransmitters. There's also two classes of presynaptic receptors. So there's what we call an autoreceptor. So, and this is different than reuptake, right? Reuptake, we're pulling the neurotransmitter back in and reusing it. Here, we're going to have, like, say this is serotonin being released here. We're going to have a serotonin receptor on the axon terminal that the own neurotransmitter can bind to as a feedback mechanism to sort of let the axon terminal know, hey, this is how much you're stimulating your target. The amount you're stimulating yourself, that's what your target's getting. And if you're not happy with that, release more serotonin. And if it's too much, back off of that. Okay? So it's a feedback. The autoreceptor is receiving its own neurotransmitter to sort of regulate that. Here's an example of norepinephrine. It's providing feedback to know how much am I stimulating my target. we can also have an axon-to-axon connection from a different neuron. And so here, this is going to be sending neurotransmitter directly to the axon terminal. So it's skipping this whole process. No synaptic potentials, no action potentials. I'm just going to go act directly on this axon terminal, right? And in that case, those receptors are called heteroreceptors. So the autoreceptor is the same neurotransmitter. A heteroreceptor, if you think about hetero meaning different, right, is a different neurotransmitter from a different neuron. And so if I want to influence this neuron's ability to send signals, this is the strongest way I can do that because I don't have to worry about input and processing and all that. I can just go directly here and influence its ability. I know this seems kind of abstract, but we're going to talk about these things in specific systems, right? When we talk about sensitization and habituation, this is all happening at the presynaptic level. That's when we get to learn input.
How does that, like, that neuron know, that axon know that it needs to tell that one to stop where he's going?
Yeah, so this says inhibitory, right? So if, for instance, that neurotransmitter, right, binds to a potassium channel and allows potassium to leave, right, what's that going to do to the inside of this axon terminal? Makes it more negative. So now when the action potential comes down there, instead of being at negative 70, maybe it's at negative 150 or something, and the action potential is not big enough to cause the voltage-gated calcium channels to open. So now, even though an action potential came down here, this neuron shut it down. Or maybe it opens a channel and allows sodium to come in and raises the level, which is going to make the action potential release more calcium and have a bigger neurotransmitter release. Okay? So it's again, it's a way to change the excitability of the system. Liddy.
In what situation do like the heteroreceptors, like when do they actually, like when do we have this work? Because they are like different.
Yeah. So I mentioned we will see this in a process, in a learning process called habituation and sensitization. So for instance, this happens with sensitization. So in sensitization, if we're sitting in here like, right, you didn't get scared, right? But if you're walking across campus and it's dark and there's no moon, and someone jumps out from behind a tree and does exactly the same thing I just did, you're going to have this big response because you're sensitized. So in that case, we know that the neurons that control that startle reflex, when you're in that environment, are making it easier for this to fire, and therefore the same do now causes a bigger response through this type of mechanism.
This is the same thing, not the same thing, but is this kind of what happens with like adrenaline or like anything like that?
Yeah. I mean, how else talk about that? That is. All right. So, some summary of this, right? We have postsynaptic receptors, which are receiving neurotransmitter as a signal from the presynaptic to the post, from one neuron to the next, right? If they're ionotropic, they're going to produce EPSPs and IPSPs, synaptic potentials, right? We can have metabotropic receptors that are going to change the excitability of this site and do complex things. We have presynaptic receptors, 2 of them, a feedback mechanism to know, hey, how much am I stimulating? Do I want to turn up or turn down my output? Right? And we can have influences from other neurons directly on that axon. Directly on that axon. All right. And then we have neurotransmitter reuptake, which is a separate process. There's actually a little transport protein, kind of like the sodium potassium pump, but it has a place to bind to the neurotransmitter, and when it binds, it flips it inside. And then there's an empty one, and then it flips it, and it keeps like pulling it in. We have glial cells out here that are absorbing neurotransmitter and breaking it down. And then we have enzymes floating around out here that can break down the neurotransmitter. All of these things are things that are happening at the level of the synapse. Good? Awesome. All right, you know what time it is. Well, this is my craziness. So we get up and do a chain of amendment. We'll just do this.
Everybody get up the next time, get moving a little bit. Alright, let's get up. Alright, first thing, keep those feet moving. You gotta keep moving the whole time. Alright, first one, we're gonna jog in place and kick heels up, tap back side. You ready? Go. 1, 2, 3.
Alright, we're getting our blood pumping. We're waking up for the last
7,
40 minutes.
9, 10. Alright, keep those feet moving. Next one, we're gonna take a giant step forward. We're gonna lower and lift back up slowly.
When you get elementary school feet.
Alright, go. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Alright, let's keep moving. Alright, third one, we're going to take 10 shots.
We're only doing one leg?
We're at the end. Alright, you ready? Alright, go. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Good job, you won the game. Good job.
Alright.
Alright, keep those feet moving. Alright, fourth one. We're going to do airplane balance pose. Hold and switch feet. You ready? We're going to switch halfway through. Alright, go. 1, 2, 3, 4, 5,
Alright,
6,
switch feet.
7,
Let's switch feet.
8, 9, 10. Alright, last one. You got 10 seconds. I'll have as many people as you can.
Alright, ready? Well, this is like a COVID thing. Everyone will do all that. We just air my body.
2,
Like, no one can run around the room and have fun.
5, 6, 7, 8, 9, 10. Good job.
Alright, here's your healthy tip.
Alright, your healthy tip of the day. Eat foods each day that are the colors of the rainbow. Red, green, yellow, purple, and blue. Colorful foods will help you get the important vitamins and minerals that are needed so you can do your best each day.
True that. Could have been so a little more enthusiastic. Alright, we're all awake now. Ready to proceed. 30, 35 minutes. Okay. So now I want to focus on the different chemicals, right? The different neurotransmitters. And like I said, they're just chemicals. They're just specific shapes here. Right? And those shapes can activate different receptors. And they activate them in this sort of lock and key mechanism. This neurotransmitter won't fit in this receptor. This neurotransmitter won't fit or activate that. Now sometimes their shape can activate multiple different receptors. They activate more than one. Sometimes they're very selective and the shape only fits in one type of receptor. So these receptors are named for the chemical that can activate them. Right? So if dopamine fits in both of these, this one would be, say, D1, and this one would be D2. They're often known like that. And this one might be excitatory, meaning when dopamine activates a D1 with this shape, it increases the excitability of that cell. And when dopamine with the other side activates D2, it's inhibitory. So dopamine can be both excitatory and inhibitory depending on which receptor it activates. It's what the receptor does that makes it excitatory or inhibitory. Good?
Receptors change shape.
No. No. So think about how the brain's organized, right? It's a mass of neurons and glial cells, and we say, okay, here's a nucleus, but right next to that nucleus are other neurons. I mean, they're not walled off or compartmentalized into these areas. And so they use different neurotransmitters so that they don't cross-communicate, right? So if one system's using dopamine, maybe the system right next to it is using a different neurotransmitter so that that dopamine, if it happens to float over into that other area, doesn't cause activation. Part of the reason we have all these different signals, it's almost like building different walls in there so that if the signal gets over into a different area, it doesn't activate that area. So these are some major neurotransmitters that we'll spend time talking about. In here, we've already talked about some of them. Acetylcholine, when did we talk about acetylcholine? What system uses acetylcholine as its neurotransmitter? Parasympathetic system uses acetylcholine. Acetylcholine is also the neurotransmitter released at the neuromuscular junction to cause your muscles to contract. So that final motor neuron, when it releases acetylcholine onto a muscle fiber, there's a receptor that makes the muscle fiber contract. We'll learn how that happens in the first lecture of the second section. It's also used in the brain as sort of an awake, alert signal, not as a wakeful signal, not alert, as a wakeful signal, right? And so the fact that you're awake and conscious in here, there's sort of your brain's being bathed a little bit with acetylcholine. It goes away when you're asleep. Okay? And it's used in some other specific areas, like learning and memory. Dopamine. Dopamine is primarily found in the central nervous system. It's not used outside the central nervous system as a signaling molecule. Some of the important things to talk about dopamine is the neurotransmitter that causes you to feel good. It's the one used in your reward system. And so if this is our dopamine receptor that makes you feel good, right? And I have another chemical that looks like this. Right? And it's called cocaine, and this part fits in the dopamine receptor. Guess what cocaine can do? Stimulate that reward center. And so almost all addictive drugs either act on dopamine here or act in a secondary way to cause your natural dopamine to be released. That's what makes them addictive. And we'll talk about that when we talk about addiction. Right? So that's a big one. Another big one for dopamine that we'll talk about before we get to addiction is that dopamine helps regulate both our thought patterns and our motor patterns. So in Parkinson's disease, again, started the second section of the course. In Parkinson's disease, we lose dopamine input to the basal ganglia, and we lose the ability to have smooth fluid movements. So you get shuffling, walk, gait, you get tremors that you're not able to control. Those sorts of motor patterns because of dopamine's role in the movement system. Dopamine's also used in the medulla and brainstem and some of those life-sustaining issues. So that means that drugs that act on dopamine can have a side effect of overdose, right? They start to affect those life-sustaining areas. All right. Endorphins are our natural pain management.